Open Access Review. Cancer Metabolism, Fasting Reset, The Keto-Paradox and Drugs for Undoing. By Maurice Israel. Eric Berg. Guy+tenenbaum. Received, December 14, 2022/revised, February 12, 2023/accepted, February 15, 2023/published, February 17, 2023. This article belongs to the section Oncology. Abstract. In tumor cells, ketolysis via succinyl-CoA, 3-oxoacid-CoA transferase, SCOT, and acetyl-CoA acetyltransferase 1, ACAT1, is a major source of mitochondrial acetyl-CoA. Active ACAT1 tetramers stabilize by tyrosine phosphorylation, which facilitates the SCOT reaction and ketolysis. Tyrosine phosphorylation of pyruvate kinase PKM2 has the opposite effect, stabilizing inactive dimers, while pyruvate dehydrogenase, PDH, which is already inhibited by phosphorylation, is acetylated by ACAT1 and is doubly locked. This closes the glycolytic supply of acetyl-CoA. 1. Introduction. Glucose and ketone bodies are the two major sources of nutrients for cells. When food is scarce, a mobilization of nutritional reserves in several organs will liberate or synthesize these nutrients. Fasting and starvation elicit the release of catabolic hormones, glucagon from the endocrine pancreas, epinephrine and cortisol from adrenals. These hormones act on different organs, glucagon for the liver, muscle adipose tissues and liver for the others. Catabolic hormones trigger glycogenolysis in liver and muscle, making glucose available. These hormones will activate the synthesis of glucose through neoglucogenesis in the liver, while muscle proteolysis provides amino acids that support the process. On the other hand, tissues responding to anabolic hormones, insulin and insulin-like growth factor, IGF, will take up glucose. Glycolysis leads to pyruvate, recovering part of the energy of glucose. Then, mitochondrial oxidative metabolism takes over. This process is energetically more efficient than glycolysis, extracting energy through the citric acid Krebs cycle, which is coupled with the respiratory electron transport chain. The citric acid cycle begins with the conversion of pyruvate into acetyl-CoA by an enzyme pyruvate dehydrogenase, PDH, controlling the glycolytic entry in oxidative metabolism. The production of ketone bodies in the liver requires the mobilization of lipid stores. Catabolic hormones trigger lipolysis, which provides fatty acids. These acids enter mitochondria through a carnitine-driven transporter, and the beta-oxidation pathway cuts them into 2 carbon units, forming acetyl-CoA. Normal liver is ketogenic, converting acetyl-CoA into ketone bodies. No ketolysis takes place in normal liver, which releases ketone bodies such as beta-hydroxybutyrate, BHB, in the blood. Ketolysis will then support the metabolism of tissues responding to anabolic hormones, primarily insulin and IGF. 3 enzymes form the ketolytic pathway, which converts BHB into acetyl-CoA. The only specific ketolytic enzyme is SCOT, the product of the OXCT1 gene. The acetyl-CoA coming from ketone bodies feeds the ketolytic entry into the citric acid cycle. In fasting, the mobilization of tissue reserves supports the energetic and metabolic demands of active mitotic tissues through the supply of glucose or ketone bodies. 2. The metabolic vulnerability of tumor cells. 2.1. Tumor metabolism. We know since Warburg, Eigenbrot and Mazurek made major discoveries surrounding cancer, 1, 2, 3, 4, that tumor cells display an inhibition of the last step of glycolysis, the pyruvate kinase M2 isoform, PKM2, providing poorly active dimers that predominate overactive tetramers. Thus, phosphorylation inhibits the enzyme. In order to overcome this bottleneck, tumor cells must incorporate much more glucose when making pyruvate and draw energy from glycolysis. However, there is a second bottleneck at the pyruvate dehydrogenase, PDH, entry in the mitochondria, which triggers the citric acid Krebs cycle. A phosphorylation also inhibits PDH, which fails to convert pyruvate into acetyl-CoA. Hence, the excess pyruvate ferments into lactic acid, even in the presence of oxygen, Warburg effect. The release of lactic acid through monocarboxylate transporters MCT1-4 decreases the intracellular concentration of lactic acid, attenuating the end product inhibition of lactate dehydrogenase, LDH. This enzyme will then be able to convert pyruvate into lactate and NADH into NAD+. The production of NAD+ is necessary for glycolysis to proceed at the glyceraldehyde phosphate dehydrogenase, GAPDH, step. Preserving this ATP-producing step, which requires NAD+, is essential for tumor survival since the other ATP-producing step at PKM2 works at a low rate, forming a first bottleneck at the end of the glycolytic pathway, before a second PDH bottleneck at the entry in oxidative metabolism. We also know that the citrate condensation reaction starting the citric acid Krebs cycle increases in tumor cell mitochondria. This will feed the synthesis of fatty acids and lipids required to form new membranes for mitotic tumor cells. 5. The exit of citrate from mitochondria starts the lipogenic pathway in the cytosol via ATP citrate lyase, ACL, and acetyl-CoA carboxylase, ACC. The product of ACC is malonyl-CoA. The latter automatically turns off the mitochondrial carnitine transporter of fatty acids and stops their degradation through beta-oxidation into acetyl-CoA when the synthesis of fatty acids is operational. 6. Which is indeed the case in mitotic tumor cells. Evidently, tumor cells need to acquire their mitochondrial acetyl-CoA to support their elevated citrate condensation. Since both the glycolytic and fatty acid acetyl-CoA entries in oxidative metabolism are narrow, tumor cells have to use ketones to create their mitochondrial acetyl-CoA through ketolysis. Liver ketogenesis forms and releases BHB in the blood. It enters tumor cells through monocarboxylate transporters MCT1-4, and then a BHB dehydrogenase forms acetoacetate, one of the SCOT substrates, while the other SCOT substrate, succinyl-CoA, comes from alpha-ketoglutarate dehydrogenase and from glutamate dehydrogenase fed by elevated glutaminolysis. The enzyme SCOT transfers the CoA of succinyl-CoA to acetoacetate, producing acetoacetyl-CoA. Then, the thiolase ACAT1 creates acetyl-CoA, which feeds the elevated citrate condensation reaction of the Krebs cycle. 7. 2.2 An essential observation illustrates the ketolytic dependency of tumor cells. The enzyme ACAT1, just downstream of SCOT, forms active stable tetramers in tumor cells, tyrosine 407 phosphorylation. This triggers the SCOT reaction and the synthesis of acetyl-CoA through ketolysis. Moreover, the tumor cells will stabilize this vital ketolytic entry because ACAT1 tetramers acquire an acetylation capability, capable of further acetylating and inhibiting the PDH entry, which is already inhibited by phosphorylation. This closes the glycolytic entry in the citric acid cycle and strengthens the ketolytic dependency of the tumor cell. 8. We also know that BHB is a histone deacetylase, HDAC, inhibitor, preserving the acetylation of histones, which induces the expression of fetal genes, for example, fetal hemoglobin in sickle cell anemia in which there is a mutation of the adult hemoglobin gene. We found that this was true for many other diseases, in which BHB induces, together with other factors, the expression of the fetal gene to replace an adult mutated gene. 9. Regarding cancer, the PKM2 is a fetal splice variant of the adult enzyme. The influx of BHB through monocarboxylate transporters, MCT1-4, 10, will form acetoacetate in the cell via a BHB dehydrogenase and feed the supply of mitochondrial acetyl-CoA via SCOT, a CAT1. On the other hand, BHB dehydrogenase converts NAD+ into NADH, counteracting the effect of lactate release that forms NAD+. This is beneficial to tumor GAPDH. An inhibition of SCOT, a CAT1 should increase acetoacetate, which would inhibit BHB dehydrogenase, end product inhibition. As a result, less BHB would feed mitochondrial ketolysis and more BHB would support the epigenetic effects in the nucleus by inhibiting HDAC, which keeps histones acetylated. This induces the expression of fetal proteins. Other ligands of the adult protein are also involved. The epigenetic effect induces a stem cell phenotype in which mitotic cells fail to differentiate. In previous works, we tested lipoic acid and hydroxycitrate in pursuit of this goal. More information can be found in refs 11, 12. We found that, respectively, these substances inhibit the citrate formation in ACL, beginning the cytosolic synthesis of fatty acids. However, tumor cells are still able to incorporate exogenous acetate, converting it into acetyl-CoA in the cytosol. Via acetyl-CoA synthetase, we can inhibit this entry with allicin, 13, or orotic acid, 14. One observation from a marine biology perspective is particularly interesting. A compound with a butanylide furan ring was extracted from a marine bacterium Streptomyces. The butanylide, 5-octylfuran, 2,5H-1, maintains the larval phenotype of barnacles, Amphibalanus amphitrite, delaying their fixation on ships or rocks and their transition to adult form. This antifouling effect seems related to ACAT1 and ketolysis, 15. Presumably, the ACAT1 inhibition caused by the butanylide leaves more BHB for epigenetic HDAC inhibition, prolonging the swimming larval life, and delays the settlement of swimming larvae and transition to the fixed adult barnacle. Compounds such as cochleomycin A inhibit the larval settlement through parallel activation of the NO-guanylate cyclase pathway, 16. A similar observation comes from vegetal biology. Plants secure their access to light and nutrients by suppressing their close competitors. They achieve this by releasing HDAC inhibitors in the rhizosphere. These inhibitors, benzoxazinoids, for example diboa, are cyclic hydroxamic acid structures, which are further degraded into more stable compounds with HDAC activity, 17, limiting the development of competitors, presumably through some similarities with indole auxins. In general, DACs, together with other compounds, NO donors, control the embryonic to adult transition for many protein isoforms. This depends, at the epigenetic level, on metabolites formed in glycolytic ketogenic monotelic fetal metabolism, or metabolites formed in adult oxidative metabolism. In mammals, a series of proteins of the aquatic fetus thus adapt to life in an environment with air and gravity. 9. Another observation relates to butanylide controlling the germination of dormant seeds after forest fires. The smoke, butanylide, carrikins, striglol, 18, signals to the seed that it can germinate. If there was fire, there is air. Presumably, carrinins mimic plant hormones such as auxin and gibberellerin. ACAT1 inhibition by butanylide may cancel the acetylation blocking PDH, 8, and turn on the oxidative metabolism. It may be interesting to determine if vitamin C, which is a butanylide, inhibits ACAT1, explaining some of its favorable effects in cancer treatment. 2.3. What about ketone diets? A ketolytic-dependent tumor will grow if fed with BHB, suggesting that ketogenic diets might be inappropriate. However, other observations show that fasting ketones would be beneficial, which may not be the case for high-fat ketogenic diets. This deserves an explanation. It has been determined that saturated fatty acids of 16 carbons and above, or trans-unsaturated ones accompanied by high-fat diets, stimulate AMP deaminase, 19. The resultant decrease in AMP will then reduce the stimulation of AMP kinase and block the inhibitory action of AMP kinase on ACC, which becomes active, boosting the lipogenic pathway. An increase in ketolysis occurs in order to produce the necessary acetyl-CoA and citrate. The increase in lipogenesis will then support the synthesis of lipidic membranes for mitotic cells. During fasting, this lipogenic boost by saturated fatty acids does not occur. On the contrary, if unsaturated components such as docosahexaenoic acid, DHA, are provided, they will inhibit AMP deaminase and keep AMP kinase active, inhibiting ACC and lipogenesis, impairing the formation of new membranes. Suppose that one inhibits the ketolytic pathway with SCOT and ACAT1 inhibitors, and eventually blocks the MCT2-4 transporters. The tumor should starve. Moreover, there are observations indicating that decreasing intracellular BHB interrupts the stimulation of NF-κB-mediated transcription and inflammation. Extracellular BHB will then activate its membrane receptors, HCA2, which are GI-coupled receptors, decreasing the inflammation further. Once activated by BHB or other agonists, such as niacin or dimethylfumarate, this receptor inhibits the NF-κB-triggered transcription of inflammatory cytokines. There is apparently an inhibition of IKB kinase, which then retains NF-κB bound to IKB in the cytosol, unable to reach the nucleus. Agonists of HCA2 such as niacin are activators of prostaglandin PGD2 release from cells infiltrating the tumor. Niacin, vitamin B3, and PGD2 elicit a skin flush. 20. In summation, intracellular BHB transported by MCT1-4 will support tumor nutrition by ketolysis and NF-κB-associated inflammation, while extracellular BHB signaling through HCA2 receptors retains the transcription factor NF-κB in the cytosol, inhibiting inflammation. This may explain opposite observations on the effects of BHB on tumors. 21-22. In the same vein, other works using breast cancer xenografts show that ketones and lactate fuel tumor growth and metastasis. 23, while other groups show that ketones decrease tumor cell viability and prolong survival using VM3 mice with metastatic cancer. 24. The MCT transporters can operate in both directions, releasing lactic acid if its cellular concentration is elevated, or incorporate BHB to fuel the ketolytic pathway. By inhibiting lactate dehydrogenase, one changes the lactic acid concentration gradient. Additionally, an increase in lactic acid in tumor cells induces oxidative stress and, presumably, intracellular acidification, which inhibits tumor progression. 25. The reciprocal interaction of tumor cells with cancer-associated fibroblasts is of particular interest. Apparently, during contact with prostate cancer cells, fibroblasts reprogram their metabolism. They take up more glucose and release more lactate by expressing new MCT4 transporters. Reciprocally, tumor cells shift toward an aerobic metabolism. They decrease their glucose transporters and increase the uptake of lactate via the lactate transporter MCT1. They adapt to use lactate for their anabolism. A symbiotic tumor stroma interaction develops and seems to depend on the hypoxia-inducible factor, HIF1. The pharmacological inhibition of MCT1-mediated lactate upload affected these reprogrammed cancer cells able to use lactate for their anabolism and survival. 25-26. 3. Metabolic features of ketolytic-dependent tumors. 3.1. The keto diet, is it appropriate? An early observation by Brunings, recalled by Clement, 27, deserves to be mentioned. In his study, patients were given a low-carbohydrate diet, which was associated with an insulin injection protocol that aimed to decrease the supply of glucose to tumors. Initially, he observed a decrease in tumor sizes, and later a rebound, while feeding patients with a high-fat diet. We now know that insulin elicits the release of somatostatin from delta cells, which decreases glucagon and ketogenesis, leading to a probable decrease in BHB and growth of ketone-dependent tumors. However, since Brunings fed patients with a high-fat ketogenic diet, a rebound of tumors took place. Brunings was not aware that somatostatin release triggered by the insulin injections was probably behind the initial reduction in tumors. 28. Indeed, somatostatin decreases glucagon and ketogenesis at the beginning of the trial. However, a late rebound of tumors probably occurs when insulin fails to maintain somatostatin release. In time, the decline in somatostatin stops inhibiting glucagon, which then boosts ketogenesis, providing ketones to the tumor. 29. In an earlier work on animal cancer models, we observed that octreotide, a somatostatin analog, decreases tumor volumes. 12. In previous publications, we tried to explain the metabolic rewiring of tumor cells by considering that tumor cells respond to both anabolic insulin and to catabolic glucagon, since enzymes such as PK and PDH remain phosphorylated, as for glucagon-mediated neoglucogenesis. Meanwhile, glycolysis and anabolism elicited by insulin affect tumor cells, displaying a hybrid metabolic situation. We have incriminated a deficient regulation of the endocrine pancreas, in which the release of GABA by beta cells failed to turn off alpha cells releasing glucagon and delta cells releasing somatostatin. The failure of this mechanism sends a dual anabolic/catabolic signal perceived by new stem with both receptors active, while differentiated cells desensitize their insulin receptors because the GABA failure fails to turn off insulin release. Concurrently, constant leakage desensitizes insulin receptors for differentiated tissues, which is not the case for new mitotic cells with new insulin receptors. Hence, these cells rewire their metabolism in the hybrid mode, while differentiated tissues produce glucose and ketone bodies to feed the ketolytic-dependent tumor mitotic cell. 3.2. Why is pyruvate kinase phosphorylated? In neoglucogenesis elicited by glucagon, the hormone acts on GS-coupled receptors, stimulating adenylate cyclase and forming CMP. The latter controls the direction of the glycolytic pathway by inhibiting an activator of glycolysis, which also stimulates neoglucogenesis, fructose 2,6-bisphosphate. This will stop glycolysis and stimulate neoglucogenesis. The glucagon receptor activates protein kinases A, pKa, then SRC tyrosine kinase that inhibits pyruvate kinase, pK. This is necessary because pK works only in the PEP to pyruvate direction. Inhibiting pK via phosphorylation avoids returning to pyruvate, allowing PEP to feed and support the synthesis of glucose. Pyruvate forms through transamination of amino acids, then pyruvate carboxylase gives OAA in mitochondria and PEP carboxykinase feeds PEP in the cytosolic neoglucogenic pathway. 3.3. How do we switch back to glycolysis? Muscle activity, or insulin action through IP3, inositol 1,3,5-phosphate, increases intracellular calcium. This activates a phosphodiesterase converting CMP into AMP, which cancels the inhibition of CMP over fructose 2,6-bis-P. This activates glycolysis and deactivates neoglucogenesis. Calcium will also stimulate calcineurin phosphatase, which dephosphorylates an inhibitor I1 of PP1 phosphatase. It will then dephosphorylate and activate PK and PDH. Moreover, calcium will increase the membrane incorporation of glucose transporters, reactivating glycolysis. 3.3.1. Selection of the acetyl-CoA source. The increase in AMP activates AMP kinase, which inhibits ACC and fatty acid synthesis, the decrease in malonyl-CoA, unlocking the carnitine transporter of fatty acid and their degradation into acetyl-CoA. Since lipogenesis is blocked, DAG does not form triacylglycerol, TAG. The increase in DAG will then stimulate protein kinase C, PKC, triggering the formation of an inhibitor, CPI-17, of PP1 phosphatase. This maintains the phosphorylation of PK and PDH by kinases, which closes the glycolytic acetyl-CoA supply, while the fatty acid acetyl-CoA source is operational. On the other hand, if DAG decreases, PKC stops forming the CPI-17 inhibitor of PP1 phosphatase. Consequently, both the glycolytic and fatty acid supplies of acetyl-CoA are operational. AMP plus DAG plus fatty acids give acetyl-CoA, the glycolytic acetyl-CoA supply is closed. AMP plus DAG minus, both fatty acid and glycolysis can provide acetyl-CoA. 3.3.2. Role of AMP deaminase. We have seen that saturated fatty acids of 16 carbons, accompanied by high-fat diets, stimulate AMP deaminase. The resultant decrease in AMP inhibits AMP kinase, cancelling its inhibitory action over ACC, which is activated. This boosts the fatty acid synthesis pathway, while malonyl-CoA turns off the fatty acid degradation into acetyl-CoA. If DAG decreases, and converts to TAG, it will not stimulate PKC. Thus, the CPI-17 inhibitor of PP1 does not form, and the phosphatase dephosphorylates and activates PK and PDH, opening the glycolytic supply of acetyl-CoA. On the contrary, if DAG increases, following the action of growth hormone, for example, PKC is stimulated, forming the CPI-17 inhibitor of PP1, which maintains the phosphorylation of PK and PDH. Since both the fatty acid and glycolytic acetyl-CoA supplies are closed, tumor cells must then use ketone bodies from the liver to create their acetyl-CoA. AMP-DAG-, fatty acid supply of acetyl-CoA closed, glucose provides acetyl-CoA. AMP-DAG+, both glycolytic and fatty acid acetyl-CoA sources are closed, ketone bodies provide acetyl-CoA, as in tumor cells. 3.4. A metabolic hypothesis for how cancer forms. Many internal or external causes can elicit cell death, triggering the mitosis of stem cells for tissue repair. If there is a chronic lesion of the endocrine pancreas affecting the anabolic-catabolic mutual exclusion mechanism, which depends on the transmitter GABA, then a dual message reaches stem cells that rewire their metabolism and become dependent of ketone bodies, while insulin-desensitized differentiated cells respond to the catabolic part of the message, providing the ketones to mitotic cells. Their ketolytic metabolism has epigenetic effects inhibiting differentiation of mitotic daughter cells, leading to a geometric increase in the tumor. 4. How to undo the metabolic advantage of tumors. The peculiarities of tumor cell metabolism that might be taken advantage of for cancer treatment have been discussed in works that consider the alterations of signal transduction pathways and metabolic reprogramming of tumor cells. 30. A common feature of tumors is to acquire nutrients, in spite of their specific metabolic alterations, and build their biomass. Associated metabolites induce epigenetic effects on tumor cells and their microenvironment. 31. In addition, the Warburg effect was revisited in relation to oxidative metabolism, indicating that mitochondria and oxidative metabolism play a prominent role in cancer, with flexible adaptation, when one controls the Warburg effect. 32. 4.1. A possible reset of the metabolic rewiring mechanism in cancer. We have seen that tumor cells avidly consume glucose, but two bottlenecks at PKM2 and PDH cut the entry in oxidative metabolism. This is similar to the process that occurs if a phosphorylation inhibiting these enzymes does not reverse while switching from neoglucogenesis to glycolysis. If one triggers neoglucogenesis through a period of fasting, in which these phosphorylations normally occur, one may try to reset the system at the interruption of the fasting period in the hope that the phosphatases will correct the anomaly, which maintains them in the off configuration. This switching mechanism, involving the role of calcium, a PDE, and a phosphatase such as calcineurin, was discussed earlier in the paper. During the fasting period, one should accept complements that preserve calcium stores and the enzymes capable of resetting the system. On the other hand, fasting forms ketone bodies that feed the tumor. We have indicated that these fasting ketones are not as bad as are those from high-fat ketogenic diets. The incorporation of complements such as green tea and epigallocatechin, 33-34, during the fasting period would inhibit MCT2-4 transporters and the ketone influx while favoring the ketone signaling action transmitted by HCA2 receptors, which is helpful. Moreover, DHA and EPA would limit the ketone contribution to the lipogenic pathway. At the end of a long fasting period, one observes some favorable cases. They might result from a successful reset, removing the abnormal phosphorylation blocking the enzymes PK, M2 and PDH. 4.2. Cutting the ketone influx. In Figure 1, we recall the inhibition of both the glycolytic and fatty acid sources of mitochondrial acetyl-CoA indicated by PK and PDH interruptions on the glycolytic entry, while a single interruption by malonyl-CoA cuts the fatty acid source of acetyl-CoA, since the tumor must synthesize its fatty acids to create new membranes. Hence, the ketolysis of BHB becomes a major acetyl-CoA supply, promoting, for example, the growth of breast tumors. Adipocytes form BHB, which enters tumor cells through MCT2 transporters. 35-36. Figure 1. Ketolytic-dependent tumor cells. Inhibitions by phosphorylation of pyruvate kinase PKM2 and pyruvate dehydrogenase PDH cut the glycolytic supply of acetyl-CoA to mitochondria. Lactic acid release occurs through MCT monocarboxylate transporters. Moreover, an acetylation of PDH by ACAT1 thiolase further increases the inhibition of PDH. Since tumors must synthesize fatty acid to create new membranes, this automatically turns off the fatty acid mitochondrial carnitine transporter and the fatty acid supply of acetyl-CoA. The carnitine transporter is inhibited by malonyl-CoA, the product of acetyl-CoA carboxylase, ACC, starting the lipogenic pathway in the cytosol. Hence, ketone bodies and beta-hydroxybutyrate, BHB, become an essential source of acetyl-CoA in tumors, via SCOT, the specific ketolytic enzyme, and ACAT1. Then, citrate exits the mitochondria and feeds lipogenesis via ATP citrate lyase, ACL, and acetyl-CoA carboxylase, ACC. By blocking the MCT transporters with indicated compounds, one would affect the lactate release, the influx of beta-hydroxybutyrate, BHB, and the supply of acetoacetate to SCOT. We indicate possible inhibitors for SCOT, lithostat or other siderophores, ferrochrome, phycocyanin, febrifugen, or even melatonin. As for ACT1, butenolides, vitamin C and others are indicated. This should starve the tumor. We suggest inhibiting the citrate condensation and supply to the lipogenic pathway using lipoic acid and hydroxycitrate, as indicated. Moreover, allicin would cut the direct conversion of acetate into acetyl-CoA in the cytosol. An essential key is that AMP kinase will inhibit ACC, which merits stimulation by acetogenin, and to inhibit AMP deaminase with docosahexaionic acid, DHA, or Galega officinalis, or metformin. By inhibiting the entry of BHB, one also stops the stimulation of NF-κB-mediated transcription, a hallmark of cancer, and cancels the inhibition of histone deacetylase, HDAC, caused by BHB. Upstream of ketolysis, one may use octreotide, a somatostatin analog, to decrease the ketogenic supply. There is, however, a paradox, since the signaling action of extracellular BHB on HCA2, a GI-coupled receptor, is to preserve, it inhibits NF-κB transcription. A prostaglandin messenger is involved. It can be strengthened with andrographylite. Niacin is another agonist for HCA2, which may help when using octreotide for decreasing ketogenesis. Green arrows, HCA2 signaling. Red arrows, BHB influx through MCT and effects. One should also control the BHB influx. Indeed, once in the cell, BHB has multiple effects. First, it feeds the SCOT pathway. Second, it goes to the nucleus, where it inhibits HDAC, favoring the acetylation of histones, which induces the expression of fetal genes. A third effect of intracellular BHB is the activation of NF-κB-mediated transcription of inflammatory cytokines IL-1B. Indeed, intracellular BHB induces phosphorylation of IKB, liberating the NF-κB transcription factor, which moves to the nucleus. Thus, if one inhibits the influx of BHB with compounds such as syringapine or cyrosingapine from Rauwolfia, 36, or others such as quercetin or epigallocatechin, 33, 34, tumor cells will starve, since ketolysis should decline, particularly if associated with SCOT and ACAT1 inhibitors. In parallel, the decrease in intracellular BHB stops the inhibition over HDAC, which will deacetylate histones, silencing fetal genes, and elicit a transition to adult genes. Hydroxamic acid HDAC derivatives, which display anti-cancer effects, are probable SCOT inhibitors, and may be able to starve the tumor. Finally, the decrease in intracellular BHB stops the stimulation of inflammatory cytokines transcription mediated by NF-κB, since NF-κB remains bound to IKB in the cytosol. In summation, the MCT transporter of BHB driven by SCOT, ACAT1 attracts this essential nutrient for tumor cells. In parallel, this elicits NF-κB transcription and inflammation, whereas HDAC inhibition by BHB elicits histone acetylation and the expression of fetal genes. By inhibiting the MCT transporter of BHB in the SCOT, ACAT1 pathway, one starves the tumor and blocks NF-κB transcription, histones are deacetylated by HDAC, favoring a shift to adult genes. Moreover, extracellular BHB activates HCA2 receptor signaling as other agonists of these receptors, such as niacin. These receptors inhibit inflammation by keeping NF-κB bound to ICB in the cytosol, further blocking inflammation and inflammatory macrophages. These effects would be mediated by prostaglandins. Niacin or other agonists of this receptor should decrease inflammation and NF-κB-mediated transcription, as adrographylide, Andrographis paniculata, 37. In conclusion, the ketone body is favorable if it stays outside the cell, acting as a signaling molecule rather than as a nutrient for the tumor. This summarizes the Keto-Paradox and explains some opposing observations on the effects of BHB on tumor cells. 4.3. Cutting the SCOT, ACAT1 ketolytic steps. Our strategy is to block the ketolytic pathway, particularly the specific step driven by SCOT and the supply of acetyl-CoA to lipogenesis, as we recently attempted on an animal cancer model. 38. Moreover, since active ACAT1 tetramers collaborate with SCOT to pull in the ketolytic nutrient, we suggest using inhibitors of both enzymes. A typical inhibitor for SCOT is acetohydroxamic acid, which was introduced by Pickart and Jenks. 39. There are many hydroxamic acid derivatives in the siderophores iron chelator family molecules, and it would be interesting to test them on SCOT activity. An association with ACAT1 inhibitors, butanolides, including vitamin C, orecholine and trimetazidine, Vastarel, discussed above would certainly be interesting to test. 4.4. Possible compounds for inhibiting SCOT. Lithostat acetohydroxamic acid is a typical SCOT inhibitor used to treat bladder stones. Another inhibitor, pymazide, 4041, used to treat mental diseases, reduces cancer incidence. Several interesting compounds are highlighted in the work of Lisanti's group, who describe potential SCOT inhibitors, the mitoketosine, 42, through their structural and binding properties. Selecting the best and least toxic derivative for animal cancer models requires collaboration with pharmaceutical groups. We can also explore the literature for hydroxamic acid derivatives given for other pathologies. This is the case for vorinostat saha or quizinostat and tricostatin, which are HDAC inhibitors, 43, that display anti-cancer properties. It would be interesting to see if they inhibit SCOT as lithostat. Another way to find potential SCOT inhibitors comes from bacteriology. In order to survive in a hostile environment such as in stomach acid or in the bladder, the bacteria Helicobacter or Proteus use their urease to make ammonia and neutralize the acidity. Drugs such as acetohydroxamic acid inhibit the urease by binding the metal, Ni, in the active center, blocking the alkalinization of urine and stone formation. Another source of compounds used to find SCOT inhibitors is the pharmacology of metalloproteinase inhibitors. Again, the hydroxamate derivatives that block the ZN metalloproteinases deserve to be studied in relation to SCOT inhibition. 44. The iron-binding siderophores of bacteria, algae and other organisms are likely the best to explore. In our aerobic environment, iron bioavailability is low due to its insolubility. Iron is necessary for the survival of these organisms, thus, they synthesize iron chelators to capture ferric iron. One category of these siderophores is hydroxamate derivatives. They are potential SCOT inhibitors and have clear anti-cancer properties. 45. We recently compared acetohydroxamic lithostat and lithothamnion from red algae Lithothamnion calcareum, 46, on the Lewis cancer model and confirmed observations on their anti-cancer properties. 38. An interesting siderophore is ferrochrome from Lactobacillus casei, which inhibits colon cancer. 47. The iron chelators desferoxamine, desferol, display antineuroblastoma effects. 48, and as such they would be interesting for future leukemia treatments. Soluble seawater marine siderophores, ferrioxamines, phytoplankton siderophore from Rhodomonas ovalis or siderophore from the marine bacterium Alteromonas luteoviolacea, Alterobactine, 49, would be interesting to test using the SCOT assay developed by Williamson, 50-51. The problem is finding the best SCOT inhibitor that would reach the enzyme without being toxic. Presumably, a hydroxamate derivative originating from the biology of siderophores or a compound related to iron metabolism would do the job. We know that the normal liver never expresses the specific ketolytic enzyme SCOT, only ketogenesis takes place in liver. Is this related to the recycling of the heme, iron recovery and bilirubin metabolism in the liver? During bacteria or parasite infections, the microorganisms try to rob our iron with their siderophores, and proteins of our immune system with better affinity for iron compete with bacterial siderophores. A colleague pointed out that melatonin has reactive groups similar to acetohydroxamic acid. We also noticed that febrifugen from Dicoa febrifuga used in Chinese medicine and its derivative halofugenone, 52, have similar reactive radicals. They both have anti-cancer effects, affinity for iron, and might inhibit SCOT. 4.5. Compounds affecting ACAT1. ACAT1 activity in association with SCOT secures the vital ketolytic supply to tumor cells by pulling in the influx of BHB. The antifouling butenolide, or vitamin C, or carrageenan and strigol from smoke, would inhibit ACAT1. Arecaline from Areca catechu is an ACT1 inhibitor, but it might have oral carcinogenic effects. Other ACAT1 inhibitors, Vastarel, could be tested in this context in association with SCOT inhibitors. The acetogenin from Anance are particularly interesting, since they have a furan ring. As butenolide, they might inhibit ACAT1 and inhibit AMP deaminase, which stimulates AMP kinase that cuts the lipogenic supply. Fungal siderophores from Felinus linteus or Felinus baumii, 53-54, are identified, and might influence SEO-TANDACAT1. Inoscavin has a furan ring, which likely affects ACAT1. 4.6. Compounds acting downstream of SCOT, ACAT1. In earlier works we showed that lipoic acid and hydroxycitrate from Garcinia were able to limit the citrate efflux from mitochondria and inhibit ATP citrate lyase, ACL, in the cytosol, which cuts the lipogenic supply. These effects are strengthened if one uses allicin or orotic acid to block the direct incorporation of external acetate via acetyl-CoA synthetase. Moreover, one can impair the synthesis of lipid membranes with DHA through the inhibition of AMP deaminase, which leaves more AMP to stimulate AMP kinase. This then inhibits ACC, at the start of the lipogenic pathway. One can reinforce these effects by decreasing the synthesis of cholesterides, using statins that are difficult to handle, or bergamottin, 55. An interesting report, 56, shows that tumors can use the glutamine entry in the citric acid cycle to form citrate and feed the lipid synthesis pathway. The role of isocitrate dehydrogenase, IDH2, is essential in this process. IDH2 mediates a reductive carboxylation to facilitate the utilization of glutamine through alpha-ketoglutarate to citrate reversible steps of the Krebs cycle and feed the citrate supply to lipid synthesis. In this report, the long-term induction of the oncogene KRAS upregulates IDH2. This facilitates glutamine utilization for lipid synthesis during malignant transformation. There is certainly a great variability among tumors and their metabolic adaptations. The presentation given in this review did not consider the different tumor situations or the magnitude of the glycolytic bottlenecks, or the degree of the ketone dependency. 5. Conclusions. With a PDH locked by phosphorylation and by acetylation, tumor cells have difficulty to use the glycolytic source of acetyl-CoA, in a way they are abnormal, as in beriberi. Moreover, the PKM2 bottleneck pushes them to increase the influx of glucose fermented into lactic acid. Tumor cells must synthesize fatty acids needed for their mitotic lipid membranes. This process automatically inhibits the degradation of fatty acid into acetyl-CoA. Hence, they become dependent on ketone bodies to make their mitochondrial acetyl-CoA. The dark side of ketolysis is the nutrition and growth of tumors, an effect opposite to a beneficial ketone signaling action, which leads to contradictory results on the effect of ketones in cancer research. This is discussed in the Keto-Paradox. We think that the specific ketolytic enzyme SCOT is a major target for cancer treatment. However, we do not think that a single compound would be sufficient for developing a metabolic treatment. The strategy is to attack each step of the ketolytic process. This would gradually starve the tumor in preference to other tissues because, unlike other tissues, tumors are particularly dependent on the ketogenic supply for their metabolism. Downstream of SCOT, a CAT1 system, attracting the ketone, lipoic acid, hydroxycitrate, allicin, docosahexaenoic acid and bergamottin should decrease the lipogenic supply. The effect of gallopine from Galega officinalis on AMD aminase is to be considered in relation to metformin. Upstream of SCOT, a CAT1, one can inhibit the ketone transporters MCT2-4 using several compounds, epigallocatechin, syringapine or cyrosingapine, while ketogenesis would be slowed with octreotide, a somatostatin analog. As for SCOT, we have still to find the best hydroxamic acid derivative in the list of siderophores. We do not think that a toxic or irreversible compound is an option, since we want to selectively starve the tumor and not affect other tissues. Suggestions could be a hydroxamic acid derivative, lithostat, lithothamnion, or even melatonin, ferrochrome or febrifugen. There are many to test using the SCOT assay, with compounds coming from the field of metalloproteinases or DACs. For ACT1, which facilitates SCOT activity, we discussed several possibilities: vitamin C, butenolide and acetogenins. Those with a furan ring might inhibit ACAT1, 57, and stimulate AMP kinase. Other ACAT1 inhibitors, such as Vastarel, Carakin, Strigol and Arecaline, should be checked for side effects. Finally, if we limit the ketogenic supply to the SCOT ketolytic pathway, it could be useful to preserve the signaling action over the HCA2 receptors, activated by agonists such as niacin. In the course of a fasting reset, aiming to correct enzymatic anomalies, one would take these compounds after checking the mixtures on animal models for possible toxic interactions. One would certainly avoid a high-fat keto diet, as explained in the Keto-Paradox. It is important to note, as we reach the conclusion of this paper, that a mixture of low-toxicity compounds, affecting each step of the ketolytic supply to ketone-dependent tumor cells, would gradually suppress their metabolic advantage and immortality, rendering them visible to our immune system and lead them to an apoptotic end. Author contributions. Following discussions with GT and EB on fasting, cancer and on the Keto-Paradox, they convinced MI that it was necessary to explain the ketone dependency of tumors, and to write this review. All authors have read and agreed to the published version of the manuscript. Funding. This research received no funding. The APC was funded by a non-profit association, Survivor O Cancer. Acknowledgments. We are grateful for discussions we had with L. Schwartz, T. Seyfried. We are particularly indebted to Guillaume Petit for his interesting suggestions. Additionally, we are grateful for the experiments performed by R. Abul Hassani to test the SCOT dependency of tumors using the Lewis cancer model. Conflicts of interest. The authors declare no conflict of interest. References. Warburg, O. On the Origin of Cancer Cell. Science 1956, 123, 309-314. Google Scholar, Cross-Ref, Pubmed. Warburg, O. On Respiratory Impairment in Cancer Cells. Science 1956, 124, 269-370. Google Scholar, Cross-Ref, Pubmed. Mazurek, S. Eigenbrot, E. The Tumor Metabolome. Anti-Cancer Res 2003, 23, 1149-1154. Google Scholar, Pubmed. Eigenbrot, E. Gerbracht, U. Mazurek, S. Presek, P. Fries, R. Carbohydrate Metabolism and Neoplasia, New Perspectives for Diagnosis and Therapy. In Biochemical and Molecular Aspects of Selected Cancers, Priestlow, T.G., Priestlow, T.P., eds, Academic Press, Cambridge, Massachusetts, USA, 1994, Volume 2, pages 311-385. Google Scholar. Schlichtholz, B., Turin, J. Goik, E. Bernacki, M. Yoskovich, K. Sledzinski, Z. Swierczynski, J. Enhanced citrate synthase activity in human pancreatic cancer. Pancreas 2005, 30, 99-104. Google Scholar, Cross-Ref. Foster, D.W. Malonyl-CoA, the regulator of fatty acid synthesis and oxidation. J. Clin. Invest. 2012, 122, 1958-1959. Google Scholar, Cross-Ref, Green version. Israel, M. Schwartz, L. Tumor cells are vitally dependent upon ketolysis, inhibition of succinyl-CoA 3, oxoacid-CoA transferase should block them. OJBS 2020, 220-226. Google Scholar, Cross-Ref. Fan, J. Lin, R. Chen, D. Xia, S. Elf, Se, Lu, S. Pan, Y. Pan, Y. Su, H. Chien, Z. Et al. Tetrameric acetyl-CoA acetyltransferase 1 is important for tumor growth. Mole cell 2016, 64, 859-874. Google Scholar, Cross-Ref, Green version. Israel, M. Genetic adaptation controlled by methylations and acetylations at the nuclear and cytosolic levels, a hypothetical model. Neurochem. Res 2003, 25, 631-635. Google Scholar, Cross-Ref. Hales trap, AP. The monocarboxylate transporter family, structure and functional characterization. IUMB Life 2012, 64, 1-9. Google Scholar, Cross-Ref. Schwartz, L. Abulhassani, M. Guise, A. Sanders, E. Steyart, J. M. Campion, F. Israel, M. A combination of alpha-lipoic acid and calcium hydroxycitrate is efficient against mouse cancer models. Preliminary results. Oncol. Republic 2010, 23, 1407-1416. Google Scholar, Cross-Ref, Green version. Schwartz, L. Guise, A. Israel, M. Junid, B. Steyart, J. M. Crespi, E. Baronzio, G. Abulhassani, M. Tumor regression with a combination of drugs interfering with tumor metabolism, efficacy of hydroxycitrate, lipoic acid and capsaicin. Investig. New Drugs 2013, 31, 256-264. Google Scholar, Cross-Ref, Pubmed. Fauca, M. Feld, A. Lichtenthaler, K. Allison. A naturally occurring antibiotic from garlic, specifically inhibits acetyl-CoA synthetase. FEBS 1990, 261, 106-108. Google Scholar, Cross-Ref, Pubmed, Green version. Bernstein, B.A., Richardson, T. Amundsen, CH. Inhibition of cholesterol biosynthesis and acetyl-CoA synthetase by bovine milk and orotic acid. J. Derry-Sy, 1977, 60, 1846-1853. Google Scholar, Cross-Ref, Pubmed. Zhang, Y. F., Zhang, H. He, L. Lu, C. Su, Y. Xian, P. Y. Butenolide inhibits marine fouling by altering the primary metabolism of 3 target organisms. ACS Chemistry Biol 2012, 7, 1049-1058. Google Scholar, Cross-Ref, Pubmed. Wang, K. L. Zhang, G. Sun, J. Su, Y. Han, Z. Lu, L. L. Cochleomycin A inhibits the larval settlement of Amphibalanus amphitrite by activating the NO/cGMP pathway. Biofouling 2016, 32, 35-44. Google Scholar, cross-ref. Venturelli, S., Bells, R.G., Camper, A., Berger, A., Von Horn, K., Wegner, A., Bacher, A., Zabulon, G., Langenecker, T., Kolbacher, O., et al. Plants release precursors of histone deacetylase inhibitors to suppress growth of competitors. Plants Cell 2015, 27, 3175-3189. Google Scholar, cross-ref, green version. Nelson, D.C., Riseborough, J.A., Flamati, G.R., Stevens, J., Gisalberti, E.L., Dixon, K.W., Smith, S.M. Carrikins discovered in smoke trigger Arabidopsis seed germination by a mechanism requiring gibberellic acid synthesis and light. Plant Physiol, 2009, 149, 863-873. Google Scholar, cross-ref, green version. Yoshino, M., Miyajima, E., Tsushima, K., Kinetics of the interactions of AMP deaminase with fatty acids. J. Biol, Chemistry 1979, 254, 1521-1525. Google Scholar, cross-ref. Cashin, Biological Roles and Therapeutic Potential of Hydroxyl Carboxylic Acid Receptors. Front. Endocrinol. 2011, 2, 1-12. Google Scholar. Okumura, S., Konishi, Y., Narukawa, M., Sugiura, Y., Yoshimoto, S., Arai, Y., Sato, S., Yoshida, Y., Suji, S., Uemura, K., et al. Gut bacteria identified in colorectal cancer patients promote tumorigenesis via butyrate secretion. Nat Commune. 2021, 12, 56-74. Google Scholar, cross-ref. Dimitrieva Pasako, O., Wong, A.C., Lundgren, P., Golos, A.M., De Camps, H.C., Kramer, Z., Tion, Y., U., B., Eskiokak, O., Edgervari, G., et al. Beta-hydroxybutyrate suppresses colorectal cancer. Nat Commune. 2022, 605, 160-165. Google Scholar, cross-ref, Pubmed. Bonicelli, G., Tsarigos, A., Whitaker-Menez, D., Pavlides, S., Pestel, R.G., Chiavarina, B., Frank, P.G., Flamenberg, N., Howell, A., Martinez-Outscorn, U.E., et al. Ketones and lactate fuel tumor growth and metastasis. Cell Cycle 2010, 9, 3506-3514. Google Scholar, Pubmed. Poff, A.M., Ari, C., Arnold, P., Seyfried, T.N., D'Agostino, D.P. Ketone supplementation decreases tumor cell viability and prolongs survival of mice with metastatic cancer. J. Cancer 2014, 135, 1711-1720. Google Scholar, cross-ref, Pubmed, green version. Lu, A., Cooper, C.R., Gao, A.M., Dinavahi, R., Mitra, A., Deck, L.M., Royer, R.E., Vander Yacht, D.L., Semenza, G.L., Dang, CV. Inhibition of lactate dehydrogenase A induces oxidative stress and inhibits tumor progression. Proc. Natal Academic, Sci., USA 2010, 107, 2037-2042. Google Scholar, cross-ref, green version. Fiasci, T., Marini, A., Giannoni, E., Taddi, M.L., Gandolini, P., De Donatis, A., Lanciotti, M., Cerny, S., Sirai, P., Chiarugi, P. Reciprocal metabolic reprogramming through lactate shuttle coordinately influences tumor-stroma interplay. Cancer Res 2012, 72, 5130 to 5140. Google Scholar, Crossref, Green version. Clement, R.J. Wilhelm Bruning's forgotten contribution to the metabolic treatment of cancer utilizing hypoglycemia and very low carbohydrate, ketogenic, diet. J. Tradett. Complement. Med. 2019, 9, 192 to 200. Google Scholar, Crossref. Vergari, E., Knudsen, J.G., Ramrakia, R., Salehi, A., Zhang, Q., Adam, J., Wernstedt Asterholm, I., Benrick, Bryant, L.J.B., Chibolina, M.V., et al. Insulin inhibits glucagon release by SGTL2, induced stimulation of somatostatin secretion. Nat Commune. 2019, 10, 139 to 150. Google Scholar, Crossref, Green version. Israel, M. Schwartz, L. Inhibition of the ketolytic acetyl-CoA supply to tumors could be their Achilles heel. Int J. Cancer 2020, 147, 1755 to 1757. Google Scholar, Crossref. Cromer, G. Puisiger, J. Tumor cell metabolism, cancer's Achilles heel. Cancer Cell. 2008, 13, 472 to 482. Google Scholar, Crossref. Pavlova, N.N., Zhu, J., Thompson, C.B. The Hallmarks of Cancer Metabolism, Still Emerging. Cell Metab. 2022, 34, 355 to 377. Google Scholar, Crossref. Kasim, S., Vucedic, M., Zdrelevich, M., Puisiger, J., Warburg and beyond. The power of mitochondrial metabolism to collaborate or replace fermentative glycolysis in cancer. Cancers 2020, 12, 1119. Google Scholar, Crossref, Pubmed. Konishi, Y., Kobayashi, S., Shimizu, M., T. Polyphenols inhibit the transport of dietary phenolic acids mediated by the monocarboxylic acid transporter, MCT, in intestinal Caco-2 cell monolayers. J. Agric, Food Chemistry 2003, 51, 7296 to 7302. Google Scholar, Crossref, Pubmed. Pornar Mohammadi, S., Grimaldi, M., Strid, M.H., Lavallard, V., Wage Peterson, H.S., Woolheim, C.B., Makler, P. Epigallocatechin 3-gallate, EGC, activates AMPK through the inhibition of glutamatidehydrogenase in muscle and pancreatic cells, a potential beneficial effect in the prediabetic state. Int J. Biochem. Cell Biol 2017, 88, 220 to 225. Google Scholar, Crossref, Green version. Huang, C.K., Chang, Ph., Kwa, W.H., Chen, C.L., Zheng, Y.M., Chang, K.G., Xu, J. Y., Hu, C. M., Li, W.H. Adipocytes promote malignant growth of breast tumors with monocarboxylate transporter 2 expression via beta-hydroxybutyrate. Nat Commune. 2017, 8, 14706. Google Scholar, Crossref, Pubmed, Green version. Benjamin, D., Roby, D., Hindupur, S.K., Paulman, J., Colombi, M., Elshemerly, M.Y., Myra, S.M., Maroney, C., Lane, H.A., Hall, M.N. Dual inhibition of lactate transporters MCT1 and MCT4 is synthetic lethal with metformin, due to NAD plus depletion in cancer cells. Cell Republic 2018, 25, 3047 to 3058. Google Scholar, Crossref, Green version. Yang, Ch, Yen, TL, Su, Cy, Thomas, PA, Shu, Jr., Jayakumar, T. Multi-targeting andrographylide, a novel NFKB inhibitor, as a potential therapeutic agent for stroke. Int J. Mol Sci, 2017, 18, 1638. Google Scholar, Crossref, Pubmed. Abul Hassani, R. Berg, E. Tenenbaum, G. Israel, M. Inhibition of SCOT and ketolysis decreases tumor growth and inflammation in the Lewis cancer model. J.J. Oncol. Clin. Res 2022, 3, 1-12. Google Scholar. Pickart, C.M., Jenks, W.P. Formation of stable anhydrides from CoA transferase and hydroxamic acids. J. Biol, Chemistry 1979, 254, 9120-9129. Google Scholar, Crossref. Goncalves, J.M., Barcelos Silva, C.M., Rivero, E.R.C., Cordero, MMR inhibition of cancer stem cells promoted by pymazide. Clin. EXP Pharmacol. Physiol, 2019, 46, 116-125. Google Scholar, Crossref, Green version. Al-Batran, R. Gopal, K. Kapotsi, Chahadi, J.J., Sulami, B. Tabatabiidakili, S.A., Greenwell, A.A., Nyo, J., Almuteri, M., Byrne, N.J., et al. Pymazide alleviates hyperglycemia in diet-induced obesity by inhibiting skeletal muscle ketone oxidation. Cell Metab. 2020, 31, 909-919. Google Scholar, Crossref. Martinez Outscorn, U.E., Lin, Z., Whitaker Meneses, D., Howell, A., Satya, F., Lasanti, MP. Ketone body utilization drives tumor growth and metastasis. Cell Cycle 2012, 11, 3964-3971. Google Scholar, Crossref, Pubmed, Green version. Pal, D. Saha, S. Hydroxamic acid, a novel molecule for anti-cancer therapy. J. ADV, Pharm. Technol, Res 2012, 3, 92-98. Google Scholar, Crossref, Pubmed. Rothenberg, M.L., Nelson, R., Hand, K.R. New drugs on the horizon, matrix metalloproteinase inhibitors. STEM. Cells 1999, 17, 237-240. Google Scholar, Crossref, Pubmed. Wilson, B.R., Bogdan, A.R., Miyazawa, M., Hashimoto, K. Siderophores in iron metabolism, from mechanism to therapy. Trends Mol Med, 2016, 22, 1077-1090. Google Scholar, Crossref, Green version. Aslam, Amen, Bergen, I, Naik, M., Hampton, A., Allen, R., Kunkel, S.L., Rush, H., Varani, J. A multimineral natural product inhibits liver tumor formation in C57BL mice. Biol, Trace Elementary Res 2012, 147, 267-274. Google Scholar, Crossref, Green version. Konishi, H., Fujiya, M., Tanaka, H., Ueno, N., Marichi, K., Ikuta, K., Akutsu, H., Tanabe, H., Kogo, Y. Probiotic-derived ferrochrome inhibits colon cancer progression via JNK-mediated apoptosis. Nat Commune. 2016, 7, 12365. Google Scholar, Crossref. Blatt, J., Steitley, S. Antineuroblastoma activity of desferoxamine in human cell lines. Cancer Res 1987, 47, 1749 to 1750. Google Scholar. Reed, R.T., Liv, D.H., Faulkner, D.J., Butler, A. A siderophore from a marine bacterium with an exceptional ferric ion affinity constant. Nature 1993, 336, 455 to 458. Google Scholar, Cross-Ref. Murray, E.M.F., Nieto, M.J., Sindelarand, R.D., Williamson, J.S. Hydroxamic acids as pharmacological agents. Kerr. Med. Chemistry 2002, 9, 1631 to 1653. Google Scholar, Cross-Ref. Williamson, D.H., Bates, M.W., Page, M.A., Krebs, H.A. Activities of enzymes involved in acetoacetate utilization in adult mammalian tissues. Biochem. J. 1971, 121, 41 to 47. Google Scholar, Cross-Ref. Mi, L, Zhang, Y, Su, A, Tang, M, Xing, Z, He, T, Wu, W, Li, Z, halofuginone for cancer treatment, a systematic review of efficacy and molecular mechanisms. J. Funkt. Foods 2022, 98, 105237. Google Scholar, Cross-Ref. Li, I, K, Han, M, S, Li, M, S, Kim, Y, S, Yun, B, S. Styropyrones from the medicinal fungus Felinus baumii and their antioxidant properties. Bio.org. Med. Chemistry Let. 2010, 20, 5459 to 5461. Google Scholar, Cross-Ref, Pubmed. Iliaguov, J., Gopal, N., Dixon, A., Chowdhury, M., Kano, S. Potentiation of anti-cancer effect by combination of mushroom extract and grapeseed extract on human bladder cancer cells. Cancer Sci, Res 2021, 4, 1-12. Google Scholar, Cross-Ref. Fiorillo, M., Pires Pages, M., Sanchez Alvarez, R., Bartella, L., D. Donna, L., Dolce, V., Sindona, G., Saccia, F., Capello, A.R., Lisanti, M.P. Bergamot. Natural products eradicate cancer stem cells, CSCs, by targeting mevalonate, Rho-GDI signaling and mitochondria metabolism. BBA Bioenerg. 2018, 1859, 984 to 996. Google Scholar, Cross-Ref. Liu, R., Liu, P., Bai, H., Ling, J., Zhang, H., Zhang, M., Hu, Y., Chao, P.J., Huang, P., Liu, J. Malignant transformation by oncogenic KRAS requires IDH2-mediated reductive carboxylation to promote glutamine utilization. Cancer Commune. 2022, EPUB ahead of print. Google Scholar, Cross-Ref. Menezes da Silva, E.L., Roblot, F., Mahuto, J., Cave, Coriadionin, the first annonaceous acetogenin with two double bonds isolated from Annona coriaceae. J. Nat Prod. 1996, 59, 528 to 530. Google Scholar, Cross-Ref. Disclaimer/publisher's note: the statements, opinions and data contained in all publications are solely those of the individual authors and contributors and not of MDPI and/or the editors. MDPI and/or the editors disclaim responsibility for any injury to people or property resulting from any ideas, methods, instructions or products referred to in the content. Copyright 2023 by the authors. Licensee MDPI, Basel, Switzerland. This article is an open access article distributed under the terms and conditions of the Creative Commons Attribution, CC BY, license, https://creativecommons.org/licenses/by/4.0. Share and cite. MDPI and ACS style. Israel, M. Berg, E. Tenenbaum, G. Cancer Metabolism, Fasting Reset, The Keto-Paradox and Drugs for Undoing. J. Clin. Med 2023, 12, 1589. https://doi.org/10.3390/jcm120415889. AMA style. Israel, M. Berg, E. Tenenbaum, G. Cancer Metabolism, Fasting Reset, The Keto-Paradox and Drugs for Undoing. Journal of Clinical Medicine. 2023, 12, 4/1589. https://doi.org/10.3390/jcm120415889. Chicago/Turabian style. Israel, Maurice, Eric Berg, and Guy Tenenbaum. 2023, Cancer Metabolism, Fasting Reset, The Keto-Paradox and Drugs for Undoing, Journal of Clinical Medicine 12, No. 4, 1589. https://doi.org/10.3390/jcm120415889.